A child scrapes his knee on a stone. A mother develops a fever after giving birth. A soldier survives the bullet, but not the infection that follows. Today, these moments can often be treated, but less than a century ago, a tiny wound could become a doorway to death. Then, in 1928, Alexander Fleming returned to his laboratory and noticed something extraordinary. On a culture plate contaminated by mold, the bacteria around the mold had stopped growing. That observation helped open the door to penicillin and to a new age of medicine. But what if Fleming had looked away? What if the plate had been cleaned, the mold discarded, and penicillin never discovered? The modern world might still exist, but countless ordinary moments could end very differently. This is a world without penicillin. One change, infinite consequences. To understand what disappears, we first have to remember the world that came before it. Before effective antibacterial drugs, doctors could clean wounds, drain abscesses, isolate patients, and hope the immune system finished the job. Sometimes it did, sometimes it did not. Pneumonia could spread through the lungs with almost nothing to stop it. A throat infection could enter the bloodstream. A cut from a kitchen knife, a factory tool, or a battlefield fragment could become red, swollen, and deadly. Surgery had already advanced through anesthesia and sterile technique, but even a technically successful operation could be followed by an infection doctors struggled to control. Childbirth carried the same hidden danger. A mother could survive labor, then become seriously ill from a bacterial infection days later. Medicine was becoming more sophisticated, but against many bacteria, it was still fighting with limited weapons. Fleming's observation did not instantly create a miracle drug. He studied the mold, showed that it released a substance capable of inhibiting several bacteria, named that substance penicillin, and published his findings in 1929. But penicillin was unstable. It was difficult to isolate, purify, and produce in useful quantities. For years, the discovery remained more promise than treatment. Then, more than a decade later, a team at Oxford, including Howard Florey, Ernst Chain, and Norman Heatley, transformed that promise into something medicine could actually use. They purified penicillin, tested it in animals, and began treating patients. The early results were dramatic, but the supply was painfully small. Producing enough penicillin required laboratories, governments, factories, fermentation technology, and cooperation across Britain and the United States. By the final years of the Second World War, industrial production had changed the scale of what was possible. A contaminated culture plate had become a global medicine, now erased that first clue. In this timeline, Fleming sees nothing unusual. The plate is cleaned. No paper is published in 1929. Years later, researchers searched the scientific literature for natural antibacterial substances. But Fleming's penicillin is not there, waiting to be rediscovered. Perhaps another scientist eventually notices a similar mold, perhaps not. Discovery is rarely the work of one person alone, but timing matters. A delay of 5 years could alter thousands of lives. A delay of 20 could reshape a generation. And while science searches, bacteria do not wait. When the Second World War begins, medicine still has some antibacterial options. Sulfonamide drugs already exist, and they can treat certain infections. But they are not a complete replacement for penicillin. In our alternate timeline, military hospitals rely more heavily on surgery, wound cleaning, drainage, antiseptics, sulfa drugs, and the patient's own immune system. More wounded soldiers recover slowly. More develop infections after shrapnel tears through contaminated clothing and soil. More hospital beds remain occupied for longer. More nurses, surgeons, supplies, and transport are needed for patients who might otherwise have recovered sooner. Would the absence of penicillin change who wins the war? That cannot be stated with certainty. Wars are decided by industry, logistics, leadership, geography, intelligence, and countless human choices. But the human cost would likely rise. The bullet would no longer be the only enemy. The wound itself would continue fighting after the battle ended. After the war, the ripple spreads into civilian life. Hospitals continue to improve, But every procedure carries a larger shadow. A surgeon can repair the body, yet infection can undo the work. A child with bacterial pneumonia has fewer reliable treatment options. A patient with a severe skin infection may face radical surgery or amputation rather than recovery. A mother with infection after childbirth may deteriorate before doctors can control it. The future of medicine also becomes harder to reach. Complex operations demand more than surgical skill. They depend on the ability to prevent and treat infection. Cancer treatments that weaken the immune system become more dangerous. Organ transplantation, intensive care, premature infant care, and many invasive procedures face higher barriers. This does not mean every modern treatment disappears. Medical progress rarely stops completely, but progress becomes slower, more cautious, and more expensive in human lives. The hospital of the future is still built. It is simply a place where doctors must fear bacteria far more than they do in our timeline. Humanity would not remain defenseless forever. Sulfonamides had already shown that bacterial disease could be attacked with drugs. In 1943, researchers isolated streptomycin from a soil bacterium. It would later become especially important in treating tuberculosis and other infections. Other antibiotics followed during the late 1940s and the 1950s, so a world without Fleming's discovery is not necessarily a world without antibiotics forever, but there is a hidden consequence. Penicillin did more than treat patients. Its successful development showed that substances made by microorganisms could become mass-produced medicines. It encouraged governments, universities, and pharmaceutical companies to search harder. It helped advance manufacturing methods, testing systems, and scientific confidence. Without that success, the antibiotic revolution might still arrive, but by another path, at another speed, and with different priorities. Another drug could become the symbol of the age. Another scientist could become famous. But every year of delay would be measured not only in laboratory papers, but in hospital beds, funerals, and families changed forever. And the consequences would not stop at medicine. More illness means more missed work, more children losing parents, and more families pushed into poverty. Long recoveries place greater pressure on hospitals and public finances. Population patterns shift. Military medicine changes. Public fear of surgery remains stronger. People may avoid hospitals until illness becomes impossible to ignore. Scientific research could also move in unexpected directions. Perhaps bacteriophages, viruses that attack bacteria, receive far more investment. Perhaps vaccines against bacterial diseases develop faster. Perhaps sanitation becomes an even greater obsession. And perhaps, because antibiotics arrive later, society uses them more carefully. Antibiotic resistance might develop differently, but that would not make this timeline better. It would simply exchange one set of problems for another, while generations paid the price of delayed treatment. Penicillin was not created by a single magical moment. Fleming noticed the effect. Other researchers purified the substance. Patients accepted experimental treatment. Factories learned to produce it at scale. Governments and scientists turned a fragile laboratory observation into medicine. That is the real ripple effect. History changes when discovery meets preparation, collaboration, and time. If Fleming had missed the mold, someone else might eventually have found another answer. The question is not whether humanity would keep searching. It would. The question is how many people would die while the world waited. One contaminated culture plate. One observation. One different future. What should we change next?